Thanks for having me today. My husband is an employee of GE, which is not particularly relevant to the content of the talk. So just an outline of what I'm going to talk about today. First, I'll start off with a little bit of background on diffusion studies, some of the parameters, design, and analysis choices that are involved. Then I'm going to talk about the key findings of diffusion studies in neurodevelopment, timing of changes, regional variation, etc. Get into a little bit of trying to understand some of the variation that exists among studies and among individuals. And then finally finish with interpreting the changes and what does it mean in terms of what's actually happening within the brain. So diffusion studies of neurodevelopment have almost all used diffusion tensor imaging. So that's what I'm going to focus on today, are the DTI results. This is likely because the advanced models and acquisitions require a lot longer time, typically double what DTI requires. So there have been a few non-DTI studies, mostly using kurtosis or neurite orientation dispersion and density imaging. And I'll touch on a few of these, but primarily DTI is what's been used in the past. And I'm going to assume you know the basics of DTI, but just so we're all on the same page, I'm going to be talking about the key DTI parameters, fractional anisotropy, which is a measure of the directionality of diffusion, sort of how ordered is diffusion, mean diffusivity, which is the average water movement happening, sometimes referred to as ADC, and then axial and radial diffusivity, which represent diffusion in the primary direction and then the secondary, tertiary directions respectively, generally assumed to be parallel and then perpendicular to the white matter fibers. So there are a couple of key choices in study design, and most of the studies are cross-sectional in nature. These studies can measure correlations or relationships with age. They do not measure change. Almost all of them talk about age-related changes. None of them are actually measuring that. They're measuring relationships between a diffusion parameter and age. So this is about 61 of approximately 70 studies that we identified. Longitudinal studies can measure change with age. They don't always. Some, like up here, we fit a trajectory to our data, which isn't actually measuring change within individuals, but then we also binned individuals and looked at how many of them had an increase in FA, a decrease in FA, or no change. So longitudinal studies can measure change within individuals, and certainly some of them do. There aren't really that many longitudinal studies using diffusion to investigate development. Also, a variety of choices in terms of analysis methods. And these can obviously impact the types of thing you're able to measure and the conclusions you make. Most of the really early studies used a region of interest approach, and here you can manually outline the regions that you're interested in, you average across the region, you get a value that you can then compare to age. Can be really labor intensive, especially if you're interested in multiple regions, you have a big sample size, really labor intensive. You can automate this, and as soon as you automate it, it becomes a little bit susceptible to misregistration. So there's advantages and disadvantages to all of these methods, of course. ROI approach also is limited to the regions you pick. So the more you pick, the more labor intensive it becomes. The fewer you pick, the more white matter you're potentially not assessing. A voxel-based method can be used to look at the white matter more comprehensively, and it's sensitive to more local changes because you're looking on a voxel by voxel basis. Also very susceptible to registration errors because it relies on the fact that everybody's normalized to the same template and this structure is in the exact same spot for everybody. TBSS is essentially a voxel-based approach that tries to overcome some of the limitations of the standard voxel-based ones by looking at a white matter skeleton and analyzing differences only on the skeleton. So it's a little bit less sensitive to registration and also deals a little bit better with the multiple comparison problem because in a voxel-based approach, you're looking at thousands and thousands of voxels. It also still has limitations. You're not measuring all of the white matter. It's still somewhat susceptible to misregistration. Tractography, pretty similar to ROI approach, but now you're using the diffusion direction information to trace out the white matter tracts. You're still averaging over a whole area, and it can be labor intensive if you're doing it manually. If you're doing it in an automated way, it starts to become susceptible to the registration issues, though less so than region of interest or voxel-based approaches. sensitive on a global level. You're assuming that the whole tract changes in a similar way, and you might not be sensitive to changes that occur only in one area of the tract. Along the tract, methods try to overcome some of this sensitivity issue and are able to look at more changes on a smaller, more local, in a more local way, but again, at the expense of susceptibility to registration errors, perhaps. And then finally, we have network approaches where you can trace out all of the connections in the brain and construct a structural connectivity network that you can analyze to look for things like efficiency and that sort of parameter. So the first 2 years of life see really marked remodeling of white matter. And this is reflected in diffusion studies by rapid changes in all DTI parameters. Rapid increases of FA, rapid decreases of mean diffusivity and radial diffusivity. This is one of the earlier studies from 2005. And you can see really rapid upswings of FA at the early ages. This is posterior limb, this is the splenium, both of them really rapidly increasing and then almost look like they're leveling off. ADC, which is the same as mean diffusivity, early decreases, again, almost looks like it levels off after about age 2. Same thing, just another example here, again, really rapid increases and a virtual leveling off around age 2. One DKI study also showed rapid increases of mean kurtosis, again, leveling off around age 2 or 3. And if you only looked at this type of data, you might think that development stops around 2 or 3 years. It certainly looks like it does. You can also look at total brain volume, which doesn't really change during childhood and adolescence, and you might conclude that there's not a lot going on. But of course, we know that there is quite a bit happening and that white matter development continues beyond this age range. It's just slower and more subtle than it is at the young age ranges. And the rest of my talk is really going to focus on white matter development after approximately age 5 up until young adulthood. You'll hear about the rest of the age range after. So what happens in this age range? Well, the most common, overwhelmingly found things are that FA increases and MD decreases during this age range. And this is a composite of most of the TBSS studies that look at diffusion changes in the developing brain. On the top are cross-sectional studies with narrow age ranges, less than about 10 years. In the middle, we have cross-sectional studies, but larger age ranges, more than 10 years. And then on the bottom, the 2 longitudinal studies that have been able to do a paired comparison to look at changes within individuals. One of the things that I think is striking if you try to look at all of them together is that there's quite a variation in the areas of the brain that show correlations with age or changes with age. So here, everything in green is the skeleton that was analyzed, and everything in yellow, orange, red, or blue, basically any color other than green, are areas that showed correlations or changes. So you can see very widespread changes in some of the studies. Almost every region they looked at is changing. Other studies find slightly more limited areas of correlations or change, often in the internal capsule, actually. So quite a bit of variation across studies in terms of where they're finding differences. And the longitudinal studies are quite sensitive and were able to show changes over very short time periods, as short as 1.5 years. The longitudinal studies are sensitive to FA changes within the brain. Another thing that's really clear when you look at studies with larger age spans is that changes are nonlinear. Still, most studies actually model the correlations with a linear fit, but any study that has a wide age range shows convincingly, I think, that the changes are nonlinear, that they continue into the 20s, but in a nonlinear way, faster at the beginning, slower at the end, reaching some sort of plateau or peak. There's quite a bit of regional variation as well. And the studies that look at this are typically tractography studies that separate into different areas and are able to assess the tract and then model a curve for each tract and compare the timing. So there have been 2 studies. This is our 2008 paper on top, the Tamna's 2010 paper here on the bottom. 2 studies that looked at this with tractography over wide age ranges, and both of them find nicely congruent findings. On top, we broke down our brain areas into early developing that are mostly developed by early adolescence, sort of intermediate developing that seem to be plateauing around 20 years, and then late developing that are still maturing in the 20s. On the bottom, they've shown this on a brain surface, where yellow are the areas that matured early, orange are intermediate, and the red are the more later developing ones. And sort of consistently are these colossal areas, the temporal pole here, for example, which seem to peak quite early, or plateau quite early. And then the cingulum, the uncinate, these frontal temporal regions, you can see in red here, are the later developing ones that really keep developing into the 20s. So there's this relatively consistent regional variation with early development in commissural, colossal white matter, and later development in frontal temporal areas. The network studies pretty consistently show increasing efficiency in childhood and adolescence. This study looked at neonates, toddlers, and pre-adolescents and found higher efficiency in the pre-adolescents, both global and local efficiency. This study, similar findings, higher efficiency in adults and young adults than in children, both global on this side and local on this side. There's a huge amount of variation, though, in the studies. So there are some of these consistent findings, but a lot of variation, particularly if you start to look at the actual FA values. So what I did here is every study that either reported curve-fitting parameters or showed the raw data such that I could basically extract the curve parameters myself, I plotted them on the same axis here and picked 4 pretty common regions that are looked at: the genu, splenium of the corpus callosum, corticospinal tracts, and superior longitudinal fasciculus. So if you look at the genu, the FA values that are actually reported in studies range from about 0.4 to 0.9, huge range, huge discrepancy. Partly because the studies are actually looking at different things. So a lot of these studies that find very high FA values are measuring only the center of the tract. They're TBSS studies, they're voxel-based studies, reporting really the central portion where there's very high FA. The studies that report lower FA values tend to be averaging across a large region, a whole tract, or an ROI-based approach, where they're averaging in the peripheral areas and FA values end up being lower. Same kind of variation in the splenium, a little bit less so in the corticospinal tracts and the SLF, probably because these don't have quite as high FA values in their centers. So you still see variation, but not as much. The second thing is, if you try to focus in on the development rates, there's variation, but I think there's a little bit of consistency in that the SLF, for example, has more consistent increases of FA than, say, the genu of the corpus callosum or even the splenium. There's a few studies here that have relatively flat appearing curves or slight decreases here. These were not significant, but it's still the best fit here with a little bit of decreasing. Whereas pretty universally, the SLF is going up, suggesting that this is a later developing region than the corpus callosum. So choice of model is going to really significantly impact the conclusions that you can draw. And on the previous slide, I showed all of the data from the various studies. Vast majority of them are linear. We know that linear is not the best model, and this, I think, kind of illustrates this. So here, this is data from my 2008 paper that I reanalyzed in age bins. So 5 to 10 years old, 8 to 13, 10 to 18, and 15 to 25. Pretty similar to what many studies look at in their data. And then this is the full 5 to 30 age range. And these were fit with a linear model, quadratic model, an exponential model, and a locally weighted averaging model, both for the splenium data and the uncinate fasciculus data. So a relatively quickly developing tract and a relatively slowly later developing tract. So sort of getting into this a little bit, I think it's pretty clear that a linear fit is not good across the whole age range. It misses the rapid increases at the beginning and misses this sort of leveling off that comes later. The linear fit might be okay for these smaller age ranges, but not great for the whole fit. The quadratic fit generally does a poor job overall in the splenium. The youngest age range and the oldest age range are U-shaped fits, suggesting a minimum FA value with higher values at both younger and older age ranges, which we know is not the case. The exponential fit, I think, does a pretty good job over the whole data set, but actually was not significant. That's what these dotted lines mean over most of the smaller age ranges. So you need a good data set to be able to fit more complicated models like the exponential. The local averaging does a fine job, seemingly at any age range. For the uncinate fasciculus, it seems to matter a bit less. The linear fits look pretty good over the small age ranges and actually even relatively okay over the wider age range. Quadratic fits also seem to do kind of an okay job, except for this 8 to 13-year age range where it's a U-shaped curve again, which we know isn't really plausible. Exponential, okay, not significant for this 8 to 13-year age range, but generally fine. And the locally weighted averaging is a little bit odd in that it shows these sort of decreases here, which we don't see in the bigger group, but generally does fine. So the message of this slide is really that your age range and your model influence your conclusions. So interpret the trends with caution. They're just trends. They're not necessarily reflective of what's actually happening in your data, particularly if you try to interpret it in the context of what happens outside of your actual fit. Here, you'd expect high FA from the older age ranges, which is not plausible. So, interim summary 1 here. DTI shows continued development into the 20s. FA increases and MD decreases pretty universally. Development is nonlinear and regionally varying in that the callosal areas mature first, the frontal temporal connections mature latest. There's both a local and global increase in efficiency in the network studies, and your model and your age range chosen can significantly impact the conclusions you draw about development, so interpret with caution. All of the studies I've shown, all of the studies out there, go look at anything, have quite a variation in development rates. So some people are going up, some are going down, some are staying relatively the same. And this is really important. First, it shows individual variation, but there's a lot of information there that we can probably use that's valuable information. And one of the things that probably contributes to that are some sex differences. Most studies don't assess sex differences in development rates. Okay, they might report a difference in absolute FA, but what I'm really interested in for this talk are differences in development rates. So some do investigate it and report it, and they find no significant differences. That's about 4 of the studies we looked at. Those that do report differences actually have pretty good agreement. They tend to find earlier development in girls than in boys. So this is an example here where there's a steeper curve of FA changes and MD changes in males than in females who have a shallower development curve. And the way they interpreted it, and most of the other studies do, is that this is earlier development in females. They're sort of developed a bit sooner, and then the males are kind of playing catch up a little bit later. Puberty could also be a really interesting contributor. There's not been a lot of work done on this yet. But there are some limited findings of puberty age interactions. So this study separated people that went through puberty early and people that go through puberty later and found different trajectories of mean diffusivity across certain areas, suggesting that puberty does have an influence on development rates. Genetics can also impact it. So this study looked at the heritability of white matter properties in the brain and found higher heritability in certain areas in adolescents, where it was about 70–80%, than in adults, where it was about 30 to 40%. So there's probably an influence of genetics there as well. And of course, cognition can play a role, even within healthy kids. This was a nice longitudinal study that looked at the rate of FA development over several years compared to reading scores in kids. And kids with higher reading scores were more likely to have bigger changes of FA in the left arcuate and the left inferior longitudinal fasciculus than kids with lower reading scores at the beginning. Also, if they separated kids who were increasing or decreasing FA, the increasing group had higher basic reading scores than the decreasing FA group. Can also look at development in disorders, for example, fetal alcohol spectrum disorder. And in this study, the orange ones are the exposed kids, and the gray ones are the non-alcohol exposed kids. So different rates of development of mean diffusivity, a steeper slope in the kids with alcohol exposure than the kids without. So disorders and other environmental factors like alcohol exposure can also contribute to the development rates. So summary of that part. There do appear to be some sex differences, with females developing earlier than males, although I think a lot more work is needed to really investigate that and pull it apart. Puberty and genetics seem to also play a role, although there's not a lot of information here yet, and there's room for more study. Development is related to cognition, and it's also altered in developmental disorders. So, what is driving the changes that are observed in these studies? The most commonly reported reasons for the changes that people see are myelination, axonal packing, axon coherence, fiber diameter, and tract volume. There are obviously other things that might contribute, things like water content, membrane permeability, and proliferation of oligodendrocytes. I'm not going to get really into these because there's more data on this side, but let's unpack some of these contributions here and see what evidence there is for them. So histology studies, particularly the famous Yakovlev and Lecoeur study, indicate that myelination continues into the 30s. That's this bar down here into the second and even third decade of life here, particularly in association areas in the brain. In diffusion, radial diffusivity is typically the reported driver of FA changes. So it's typically decreasing alongside the FA increases. And we know from animal studies that RD is sensitive to myelin. So some evidence sort of in favor of myelin as a contributor to the observed changes. There's also some evidence not in favor of myelin as a contributor. This is a study that looked at diffusion parameters and magnetization transfer ratio. And in FA, they saw almost every tract showed significant positive correlations of FA with age between 7 and 14. Whereas magnetization transfer ratio showed no correlations with age. So what they suggested is that because MTR is also sensitive to myelin, this suggests there isn't a myelin change happening and that it might be something else that FA is sensitive to. For example, axonal packing or axon coherence, which FA and radial diffusivity are also sensitive to. So what about axon coherence? Well, the NODI model separates out a neurite density index and an orientation dispersion index. And the orientation dispersion index supposedly gets at the idea of axon coherence. And there have been 3 studies published, and I saw another talk yesterday, so 4, looking at NODI parameters in development, and all of them report positive relationships between NDI and age, and no significant relationships with orientation dispersion index. So it really suggests that at least in this late childhood, adolescent age range, axon coherence is not a major contributor to development. Axial diffusivity can give us a little bit more information. Now, most studies don't find a lot of change in axial diffusivity. Some show slight decreases. This is some of our data here, just a really slight decrease, or no change, okay, over the childhood, adolescent age range. But there are a few studies, found 4, that report increases of axial diffusivity, or here called parallel diffusivity, during adolescence. Interestingly, it occurs most commonly in the later developing tracts. So the cingulum, the uncinate fasciculus are where these are most often reported. And the studies suggest that perhaps this is related to fiber diameter or to axon straightening. And axon straightening is something that is seen in development in rats, although it's still a little bit unclear what's happening in the humans. And finally, tract volume. White matter volume increases to about age 35, and this can contribute to partial voluming effects because if you have a smaller tract, partial voluming on the edges is a larger proportion of the tract than in a large tract where partial voluming is not going to contribute as much. So partial voluming would lower your FA values, and if tract volume goes up, you would see an increase in FA. But a few studies have looked at it, and the volume changes really do appear to be a separate process. So one study found different trajectories of tract volume versus tract FA. A couple of other studies found that the volume changes were not correlated with diffusion changes. And in our study, we statistically controlled for volume changes and found that it didn't change our diffusion findings. So what's driving the changes? Well, I think there's good evidence to show that it's likely somewhat influenced by myelin, that it's somewhat influenced by axonal packing, and it's probably not driven largely by axon coherence. It's kind of unclear what the role of fiber diameter is here, and I'd say it's very unlikely that it's tract volume changes that are driving the diffusion changes we see. So overall, we see nonlinear FA increases and MD decreases that continue into the 20s. The callosal areas mature first, frontal temporal areas mature last, and interpretations are really limited by study design and analysis choices, and I think this is very important to keep in mind when you're trying to understand what's happening. Things like sex, puberty, genetics, cognition all affect development rates, and likely drivers are myelin and axonal packing. Unlikely drivers are tract volume and axon coherence. So in conclusion, diffusion imaging has revealed a lot about brain development, and there are some consistent findings across studies. But there's a really big need for larger and longitudinal studies to clarify things like sex differences and relationships with cognition. And then there's also a need for new models and acquisitions that can help us clarify what is changing in the underlying processes. So finally, I'd just like to thank my funding sources, people that have contributed to data on the slides, and my lab, and finally, thank you for your attention.